All right, so yeah, so here I'll turn over the control of the screen to Banisha, and thanks for being here.
Thank you. Thank you for your elaborative introduction. Now I'm sharing my screen. Yeah. Yeah, so is it visible?
Yes, I see it.
Okay. So today I'm presenting my doctoral work that was carried out at the University of Delhi, India. So I tried to understand the different aspects of floral nectaries across angiosperms, and in particular family Solanaceae. So I would like to give you a tour, like how floral nectaries look like, how they are phylogenetically distributed, and what are these specific trends that floral nectary shows. So the title of this presentation is like Evolutionary Trends of Floral Nectar in Solanaceae. So to begin with, you can see here that there are many angiosperm flowers, and most of the angiosperm flowers share one particularly interesting feature, that is the presence of floral nectar. Now, floral nectar, I would like to give you a brief introduction that floral nectar is a specific organ which is destined to produce an important reward, which is nectar, and by which a flower has been able to establish its relationship with the pollinator. So it is a deciding component because it influences the plant-pollinator interaction. One particular feature of nectary, which makes nectary a trait of interest for evolutionary studies, is that the position of nectary is not fixed within a flower. It has kept on changing across angiosperms, across different clades. And that's what it makes an important or like in a very amazing trait of interest for ego studies. So my perspective from of studying floral nectary is purely evolutionary development thing. So this was the basic introduction of floral nectary. Now I wanted to show you how nectary looks like and where are the specific positions, like how does it look like, and then where it is actually within a flower. So because I'm saying that this is a floral nectary, it is associated with flower whorls only. So the next slide shows you the different kind of petal nectaries. Now, if you see this flower, it's a very simple flower. When I did a stereo microscopic analysis, I could see a small lobe-like structures on the base of the petals. And this is a scanning electron micrograph of this petal nectary. And this structure, which is an outgrowth on the base of petal, its only function is to produce and hold nectar. So this is one form of petal nectary. The structure may vary, may not vary within a clade. This is the another form of nectary that you can see that this is another flap-like structure. It's not tubular. This is also in one of the Raninkelacea species, because most of the work that I'm presenting today is published, but there are few slides which are still not published. But just to give you a highlight that how does it look like, I have put these pictures into this presentation. So this is a modified, petal modified nectary here. It is an Eranthis species again from Raninkelacea. And this whole petal has become a nectary modification structure. And this, you can see that this is a very flat structure which holds nectar inside. And this one is the Aridis odorata. It's an Orchidaceae species. Here, petal has been modified into form of nectar spur. So this is a spur, which is a green-like structure and holds nectar in it. So this is the range of variations that you would find across different species in petal nectary. So if it is associated with petal, we say it as a floral petal nectary. And when it is associated with the gynoecium, we say gynoecial nectary. This is a model system, a petunia flower. And when I scanned the electron, gynoecium, I found out that there is some triangular structure or a kind of a disc which is present at the base of the gynoecium. So this is how a gynoecial nectary looks like. So if you will see here, this is a Brassicaceae flower, and this is a nectary lobe, which is alternating with this filaments. So because it is present in the stamen wall of the flower, we called it as a staminal nectary. So nectaries are present everywhere you imagine within a flower. So I have shown you the petal nectary, the gynoecial nectary, staminal nectary. The other form of nectaries are staminodal nectary. Now this is a trolleus flower again, and inside this flower is a world of stamens and staminodes. But here, when you see the structure, it is a staminode of this flower. And at the base of the staminode, it's a secretory space here. So this is a staminodal form of nectary. The other staminodal form of nectary I've seen here in the Xanthorrhiza species, where this is a staminode in the flower. And if you see here, here, the nectaros, the stomata-like structure in the center, this is a nectar secreting structure, meaning that they are present also on stigmas, the styles, everywhere, like you can find anywhere within the flower. So when I started my work in my PhD, I had this latest information from Bernardello et al., 2007. What he did, he mapped different positions of nectary onto the APG tree. I think it is an APG tree, phylogenetic tree at the order level. And he mapped the position of different kind of nectaries present across these orders. So what he inferred from this phylogenetic tree, he said that in the basalmost clades, the nectary is restricted to the outer walls of the flower. And as you move from these anaclades to the core eudicots, and within core eudicots to the highly nested clades, this nectary has been shifted to the stamen position, and within the highly nested clade, it has been shifted to the gynoecium region. So what we called is this shift, that across angiosperms, there has been shift in the position of nectary from outer walls to inner walls. And this shift, we call it as an acrocentripetal shift. This is what the latest information I had from 2007, which supported the earlier Fahn's statement that nectary has shown acrocentripetal shift across angiosperms. But there were few questions that I wanted to answer, or with what, like I was looking for some these answers. For example, I had a question in my mind that if the position of nectary, whether it is fixed within an order or it varies. If it varies, then what kind of other nectaries you can find? And each time when the position is changing from molecular perspective, is it like that a different molecular regulator is recruited for this different kind of nectary formation, or it has happened once in the ancestor and it remains the same? And how frequently these different kinds of nectary arose within the angiosperms? There were these many questions in my mind. So I wanted to answer. I was looking for the answers. And this tree gave me a boost to the furthermore analysis, which is that what I did. So before I started my wet lab, so I tried to understand. I did a literature survey of floral nectaries on this web, this Google web and everywhere, and tried to do a meta-analysis of things. So I tried to incorporate information, whatever was available for all 416 families, which is in under APG4 phylogenetic tree. I constructed the tree using the NuVic format, and I tried to map the different kind of nectary positions onto this tree using the fig tree software. So for each nectary type, I have given a different symbol. For example, if you would see that the squares are different kinds of stamen nectary, because position of nectary also varies within the stamen wall. It can be intrastaminal, staminal, or this extrastaminal or interstaminal. For example, the triangles that I have used, it's for outer walls of the nectary. And this circular one is for the gynoecium associated nectary. So I've used these symbols to understand the phylogenetic distribution of floral nectaries across the angiosperms, to understand, to infer few answers so that I could design my objectives further. So this is how a tree looks like. This is a APG phylogenetic supported classification of this tree, and we have 416 families in it, and all the symbols showing that within this family, that information I got from different genus or species, I mapped these positions in this across the tree. And I also calculated how frequently the different kinds of nectary arose within the angiosperms. Now, to like this work, how now I wanted to just elaborate on this tree. For example, if you would see that this is just a section of this monocot clade, and this is a monocot clade. So most of the families that it is coming under the monocot clades, showing this yellow kind of symbols, mostly in, like they are mainly in many families. If you would see, you would find occasional presence of other types of nectary. So it helped me to understand, to resolve that there, I could, evolutionarily speaking, I could say that there might be one transition event associated with this septal kind of nectary here, and that's why all its descendants harbor this kind of nectary. And wherever you find other than septal nectaries, it is an independent event in the family. So if I'm saying that there is a one transition event, molecularly speaking, I would say this, there would be one of like a regulator which has been recruited only once in the ancestor of monocots, and therefore they are responsible for septal nectaries across monocots. Similarly, if you will see the rosettes clade, you would find that most of the members here are with the, you know, these squares, different squares, colored squares. These are actually the stamen associated nectary. And this rosettes clade has families which is mostly with the stamen kind of nectaries. So somehow with this analysis, I could understand that where could be how many transition events are associated with different types of nectary, and developmentally and molecularly, I could understand that where could be the where could be the different regulators, whether the similar regulator is responsible for a different for the same type of nectary, or there has been a multiple events of transitions happen. So this is how we resolve some answers, like some queries of ours, and concluded that floral nectary is a phylogenetically widespread trait that has been gained and lost on numerous occasions. There are multiple transition events that have contributed to instances of non-reproductive nectary types than reproductive types. Reproductive wall nectaries appeared more frequently in the across angiosperms, and we also supported the Fan statement that there has been a acrocentripetal movement of nectaries. And we could also see the developmental aspect. We could also highlight that there are few genes that have been co-opted for nectary formation. So this is what we analyzed from this tree. Now, because we could see that each family has a different kind of a nectaries. So there were few questions that we wanted for a PhD to focus on one family so that we could understand the morphological variations present or not, or we could also relate it to the molecular reasons behind the different kinds of nectary presence in a family. So we decided to take this one family, which is a Solanaceae. There are many reasons to select this family. First, when you wanted to understand from the evolutionary point of view, you should have a fully resolved phylogeny. So we had resolved phylogeny by Olmsted et al. And Sarkinen et al. So that we could map the distribution of different nectary types, and we couldn't, and so that we don't, spend energy on phylogeny. So this was the first reason that I chose Solanaceae as a model system. Second reason for choosing this system was that the flowers within Solanaceae, the floral forms, they are like of different types. The question that arose for working, like for choosing a Solanaceae, is that whether a nectary changes with different floral forms or different, with the floral symmetry or not. So that's why Solanaceae was a good was a choice of our study, because you would find that you have a bilabiate form of zygomorphic corolla forms, then you have trumpet shaped corollas, then there are strict tubular form corollas, you have campanulate form physalis, and then you have rotate forms. So Solanaceae covers a range of floral forms to understand whether the nectary varies with respect to the corolla forms or not. So we did a sampling according in a way that we tried to cover most of the tribes, so that we can speak of, or we can translate our knowledge into an evolutionary thing. So we tried to cover the different most of the tribes that were there in India. So the collection was purely from India. So what we observed, that a structure which appears as only a disc-like, as you would see in literature, you would always find a word if the Solanaceae has disc-like structure. After that, there is no information. But when I did a phenotypic study of nectary using anatomical and morphological study by scanning electron microscopy and anatomy by resin sectioning, I could categorize nectary into various forms. I found that the nectary can be categorized into the lobed and annular forms. Within a lobed form, it has 1, 2, or 5 lobe nectary forms. Within annular, I could categorize in the cushion and ovarian form of nectary, and within ovarian, I would say that there is a change in the position, which is lower and upper ovarian nectary type. So here I will show you that how does a one-lobed nectary looks like. This is Physoclina prelata species, and this is the gynoecium. So all Solanaceae species have gynoecium-related nectary, but the nectary differs in morphologically. So if you would see the section, this is only a lobe, like this is only a place where you would find these stomatas. The stomatas here on the nectary, we call it as nectarostomata, because they are destined to exude the nectar. So this is one of the kind of a marking to show that it has a nectary. So this is a only structure, only a kind of a only structure where it has shown nectarostomatas in a whole this disc. And anatomically, you would see that there is this kind of a lobe is a nectar secreting structure, and opposite to this, it is completely absent. So we call it as a one-lobed nectary. So the other form of nectary that you would see is two-lobe nectary. So these are the species that I studied showed two-lobe nectary. Now you see this is a Schizanthus species. So so here the nectary structure is as at the two opposite junctions of the carpel. So this is how two-lobe nectary looks like. So this is a Petunia species, and here are the two opposite lobes of the this nectary. So this type of nectary, we could call it as two-lobe nectary. So then there comes the five-lobe nectary. If you would see that this is a Brugmansia species and these are the Datura species, it has 5 lobes in the nectary disc. And these are the nectarostomata containing regions or nectar secreting region. So we call it as a 5-lobe nectary. So within a lobe, we found 3 categories: 1, 2, and 5 lobe. So then next comes is annular nectary. Annular nectary is cushion type here in cestrum, because if you would see that this is the carpel, here the disc, this annular nectary is completely differentiated from this carpal region. So we call it as a cushion kind of a nectary in cestrum species. Then this is an ovarian kind of nectary. Now, if you would see this, there is no differentiation of nectary and carpal region. Here, the base of the gynecium has turned nectariferous. That's why we call it as ovarian lower type of nectary. And it has been seen in many species like Lycium, Hyoscyamus, Nicandra. And this base of this, gynecium is full with nectarostomatas. So that's why we call it as ovarian lower type of nectary. There was one interesting the other species that shared the same ovarian lower type of annular nectary is Vidania physalis species. There is one interesting feature that we saw in capsicums here. There is no distinguishable region, not nectarostomatas in it. What we found is that nectarostomatas has been shifted from lower to upper position here. And if you would see this flower, you would find that nectar is being accumulated at the top layer of the gynecium. So there has been shift in position in capsicum species only, and that's why we call it as upper ovarian type of annular nectary. Then there were also some control genus like Solanum, where nectary is completely absent and you cannot see any kind of nectarostomata or nectar secreting region. So it acted as control for for us. Then after morphological observations of different kinds of nectary, so the question that we raised is that different kind of nectary that we have seen, how they are organized within a flower, and does it have any correlation with the symmetry of the flower or not? So what we did, we constructed a floral diagrams of all the species that we used in our analysis, covering most of the tribes except two which is not found in India. So what we have seen here in floral diagram, so these are, these are the sepals, petals, and stamen and carpel thing. But this blue thing, which is lining the gynecium, shows the nectary. So what we have observed in most of the members of Solanaceae, that lobed kind of symmetric type of nectary was always associated with the zygomorphic flowers, while this annular kind of nectary was associated with actinomorphic flowers. This is what we found kind of a correlation between asymmetry, the zygomorphy or actinomorphy, with different kind of nectaries observation. So there were various traits that we measured for nectary analysis. For example, we saw what kind of a structure it is. So these are the ancestral reconstructions for 4 traits. That is for the type of nectary, for the nectary position, for this floral symmetry, how this floral symmetry is behaving, and for also the cuticle thing. So we did some histochemical tests, but I'm not showing it here. So if I combine all the information and present it in one, one picture. So this is the picture that explains everything that I have inferred from my study. So if you see these, this is the, tree, phylogenetic tree showing the, the basal-most clades, and these are the advanced clades or the late evolved clades. So if you would see here, so this is representing the type of nectary. The blue one shows the lobed nectary, and this one shows the annular kind of nectary. Most of the nectary position, we say it from molecular perspective that it is receptacular in origin, except the capsicum, which has been shifted. So we said it's an ovarian kind of, position. And this is the floral symmetry. This represents the zygomorphic flowers, and this is, the actinomorphic flowers. And we try to associate all these traits with the different pollination syndromes. So what we observed here, the lobed form of nectaries with these zygomorphic flowers, that they are correlated with, long-term pollinators. And this annular nectary is always associated with this bee-pollinated flowers. This is what I inferred from this observation that how nectary relates to different pollination syndrome. So it is one of the pollination syndrome. And this is, we translated our observation that how the different kind of nectaries have been evolved from, because two-lobe nectary is the basal form of a nectary. And there has been, and the annular type of nectary is the advanced forms of nectary. So this is what is the Solanaceae trait evolution. But now I took this study to the next level where I wanted to understand that, we observed that there are so many morphological observations present within the Solanaceae. And there has been a, change, there has been absence of this nectary thing. So what could be the molecular reasons why the structure is so flexible? It means that the molecular machinery responsible for different kind of variations present and then absence of this nectary, there should be flexible molecular reasons. So what I have a background knowledge from molecular thing is that this is a study by Baum et al. 2001, where he explained that crab's claw is a gene which is responsible for nectary formation across pore eudicots. This creates the nectary which is associated with gynecium. So CRC is a crab's claw, and the gene, it is present as a single copy in most of the families. So with this CRC as an information as a lead that somehow how CRC is related to diverse Solanaceae nectary types and how it is responsible for change in positions from, from lower to ovarian type of nectary. So we wanted to understand the role of this gene in Solanaceae family. So this was the NCBI information available to me. So where most of the time the gene was not annotated for us. It was a very weak database for Solanaceae studies. Either the gene was not annotated or it was it is a crab's claw like. So it has not been confirmed as crab's claw. But then we make use of, this Sol Genomics Network database. This is a very nice database to work on Solanaceae family. When I did my analysis, there were 14 genomes published. Some were complete, but few were draft. But I was able to infer information from this genome. So I used the Sol Genomics Network database, and what I did. So I used the annotated copy. There is a crab's claw A gene, which is which has been confirmed for their expression in Solanum lycopersicum, which is without nectary. So I used the sequence of this gene and tried to do the blast analysis on all the genomes available to me on the database. So I found that this gene is present in all the genomes that were available. The problem was that it was not annotated, but we followed some bioinformatic pipeline and dig out the gene. So there was one crab's claw A gene for all the species available on the database, and then there was a crab's claw B gene. It means that there are two copies which were present in Solanaceae lycopersicum, and both the genes, when I tried to identify the sequences, the homologous sequences on the database, I found for all the species the two type of CRC genes present. Like there are paralogous pairs present in the data in the genome of the Solanaceae, except the Nicotiana tabacum, because it has 4 copies because of tetraploid genome. So instead of one copy of CRC gene, which is present in almost all the families of pore eudicots, Solanaceae came up with 2 copies of CRC. So again, we thought of that there must be some role of 2 CRC copies available in the genome or not. So what we did, we took the sequences of both CRCa and CRCb and tried to construct the phylogenetic tree within Solanaceae using Vitis vinifera as a root sequence, and also across angiosperms. So what we found out that these CRCa and CRCb are sister copies, and it is specific to Solanaceae family only. So which is what saying that there must be some kind of an ancestral event happened at the node of Solanaceae. That's why exclusively in Solanaceae. That's why that most of the members have have 2 copies of CRC gene. So again, we mapped both the genes like CRCa and CRCb on all the genomes available on the database, these 5 genomes were completely sequenced, so we could see that the position of the gene on the chromosomes, on different chromosomes, and these were scaffold level assembly, so we could only locate the scaffolds. But it confirmed our observation that there are 2 types of copies. There are 2 copies present in the Solanaceae, and these are paralogous copies, and there has been some ancestral duplication event occurred at the node of Solanaceae. Now the question is that if CRCa copies, there are 2 copies present, so how conserved they are across evolution. So we did a microcentenary analysis. So this is just to show you that the left side shows you the 150 kb gene, the gene construct. Each arrow represents different genes, and the left is for like reverse orientation in different orientations. But when we mapped the presence of CRCa gene, we found on all this, on all the genomes studied. So indicating that there is an important role of both the copies. And this is what we did. That's why we did intensive bioinformatic analysis to support or to understand that how significant the 2 copies are for nectary formation. So this was present across all the genomes here when we did centenary analysis of CRCb and also for CRCa. So this red arrow you would find everywhere on all the genomes. This indicates the CRCa gene. So after this, we tried to understand the role of segmental duplication. So we did a gene block kind of analysis where you have this as a genome, and it is a gene block of CRCa and CRCb, and we found that that this red represents the gene, Crab's claw gene, and it is present on both the copies. Both copies are present in the genome. So what we inferred from the analysis is that there has been a segmental duplication in this Solanaceae family. This is again a kind of the analysis to support that whether there is a segmental duplication or local duplication. The analysis shows that there has been a that this gene has been retained in all the genomes. So it again indicating its importance for the family. So this all bioinformatic analysis was only to to concrete this thing that Solanaceae has 2 copies. It arose as a result of segmental ancestral duplication, and because it has been retained in most of the genomes, therefore it is significant to study to carry another further expression studies. So we concluded from the analysis that phylogenetic analysis that concludes that several sequences that were annotated as CRC or CRC-like or YABI, because CRC is a part of YABI gene family. So they all belong to a monophyletic clade with 2 subclades at CRC and CRCb. There has been an ancestral segmental duplication event in the common ancestor of Solanaceae, which is responsible for the origin of CRCab paralogs. And the microcentenary analysis shows that CRCa and B genomic segments underwent lineage-specific changes, which may result in differential retention of CRCab homologs across Solanaceae. So I am completing my talk here, but there are we got some leads from this work, and this work is still being continued in this family also and in other families as my postdoc work. So I thanks to all my to my PIs and my other advisory members, and also to the funding agencies who has been who has supported my work in my PhD also, as well as in my present postdoc also. Thank you.
Great, thanks a lot. That was a very interesting talk. I know a lot more now about nectaries than I did before. So if anybody has any questions, you can type them in the chat and I can read them out, or you can just raise your hand or say that you have a question. And Stacy, looks like you have a question.
I have a couple questions. I'm always fascinated by nectaries, and I don't know, but I don't know that much about them. And I remember my student Chelsea and I spent a bunch of time trying to find the nectaries in Physalis. And so it's like, oh, good. Someone else has looked. We started to think maybe they're not there. Like we could see a colored ring, but I couldn't. So I have a bunch of questions for you. One is, for the nectiferous stomata, do they look any different from, like in cross section when you see them? Is there a way that you
They're
know?
always open. They're always open.
They're always open. Okay. Okay.
That's what they make. That's a difference from the other stomatas. Like we have stomatas everywhere for like gas exchange and everything, but they keep open and closed. But nectar exuding stomatas are nectarostomatas, and they're always open. And many times you would find the pores occluded with, like they're filled with secretions.
Okay. And so
I have not
oh, go ahead. Sorry.
So I have not shown a very magnified image of nectarostomata, but if I would show you, so one nectarostomata in a higher magnification, you would find several granules, filled with the spore.
Okay.
So that's a peculiar feature of nectary thing. But all nectaries do not have nectarostomata.
Right. Some can do it with hairs, right?
Yeah, so it depends. It depends. So that's why it is difficult. Solanaceae was easy system to work on nectaries, because I wanted to cover from evo-devo perspective, because I also wanted to understand how the differential expression of genes is responsible for different positions, because it is happening. It is happening that it is changing its position within a family, and that's what it impressed me. So I have my ongoing projects, because I had several hypotheses when I finished my PhD, but I'm now in my postdoc thing, so I have these projects where I am doing this work, but I have not got published, so probably I will present next time.
So just to clarify, so in Solanaceae, nectar only comes out of stomata. There are not nectiferous hairs. Okay. Because see, Physalis has all these big fluffy hairs, and I thought maybe it's coming out of the hairs since I can't find where it's coming out of on the nectary. My other question was, how a lot of Solanaceae have different color, like in Nicotiana, it's like an orange band. Is that, can I just take that? Like if you have a fresh flower and you open it up and you see a colored band, does that always coincide with the nectary? Like if I again, Physalis made me such a headache. It had the colored band, but I couldn't see where the nectar was coming from. We couldn't measure any nectar. I think it was just so small, so little nectar, but it did have the orange band. So is that sort of diagnostic, I guess, is my question of their being?
People consider it as a taxonomic character to characterize it, but it may be related to pre-pollination and post-pollination events. I'm not sure of it, and I don't know the physiological reason behind it. So I did not take this observation in my analysis. It may be or may not be.
Okay, cool. Well, thank you so much. It was really interesting.
Thank you so much, Stacy.
All right. Pat, you had your hand up first. So got a question?
Hi, thank you. Yes, I also didn't know much about nectaries, and I really appreciated your talk. I know that you've been looking at these crab claw genes, but in many cases, they're in species that don't have nectaries, right?
Yes.
And
yes.
So have you done comparative studies with like a closely related species, like capsicum versus lycopersicum? Have you looked at gene sequences and expression of plus and minus nectaries?
It's a very interesting question, and I ended up my PhD with this question, because I could only work on Nicotiana, because my initial thing was that whether both the genes are present in the species or not, because I have dig out all the genes bioinformatically, but I want to validate the expression of the genes. I took observations on Nicotiana, but I did not extrapolate it to the capsicum, which is my ongoing publication, because I have not done this study where it has been shifted in position, because I am really, I have some clues, but I am not sure of it, how this CRC genes will behave. And I have not done any kind of transcriptome analysis for this nectary region in the upper position, because it is on the ovary position, like the capsicum nectary has been shifted to the upper part. So it is very difficult to delimit the genes from ovary, because what if I'll do like the sequence thing, it nectary genes and ovarian genes will be mixed pool of something. So I have not done the analysis on capsicum yet, but that would be very cool. I think I need another postdoc position for it, because it is a big kind of a work to do furthermore.
But there's probably something known in tomato already.
Yes, we know it tomato. Tomato doesn't have nectary, and it has an annotated CRCa gene. So we know that in the higher nested clades, the gene has got, there has been change in the expression, because in both the genes are expressed in different regions of ovary.
Oh. Okay. So some work has been, the foundation has been laid.
Foundation has been laid only in Nicotiana, where they show the expression of CRCa and B via in situ hybridization, but they have not done it in the capsicum species. And in Solanum, they have just a very preliminary analysis on it, but they have not showed in a concrete way that it is actually expressing.
Okay, thank you.
All right, Chris.
Awesome stuff, really interesting, and this turns out, based on what you were just saying, that this maybe is a decent follow-up. So I study Solanum here at my school, and mostly Australian spiny Solanums, and where I think we're up to like 15 or 18 that we've now confirmed to have extrafloral nectaries, right? And so these spinies are like oozing this stuff all the time, and it's still model, and it's coming out of mostly the backs of the corollas and also the backs of leaves, but also some other places. So we're doing some work here to look at the constituents of that stuff and see what kind of amino acids and proteins are there, and try to align it with potential ant mutualisms, right? But I wonder, like, thinking about this, if even the floral nectaries are stomatal in origin, right? Then I'm sitting here wondering, like, whether or not the sort of on-off switch is really similar for whether they're inside the flower or outside the flower, right? If it's all nectariferous stomata, what are your thoughts on that?
Okay.
Sorry.
So the thing is that in Solanums, we have extrafloral nectaries, and we have CRC expression there also in the extrafloral nectaries. So what is that something which is on off, like within the flower or outside the flower, is some downstream regulator of CRC gene? Possibly. Yep. And we don't know molecular reason, because we are up to the point where we know that CRC is a master regulator, but what comes after CRC, we don't know. And that's what makes this structure flexible.
Yeah.
And it's a group of, this belongs to a Yabi gene family. So this Yabi gene family has 5 gene subfamily members, and they all have a role in abaxial and adaxial expression of, in leaves, in flowers, in ovules. So somehow, if we will try to understand the different member roles, maybe there could be some other gene family members within the Yabi who is playing a role somewhere like that. We have no idea. I started my work from the scratch. I came up with this observation. Then I was looking for some postdoc who could support my work. Eventually, I got in Tel Aviv University, but then I'm continuing my work on other families because I was also interested by the time. But I have changed my focus from here to floral color. But seriously, I have no idea that what caused these on and off switches, and this what makes nectary an interesting thing.
It's interesting that it may be this, it's CRC either way, we assume, right? Which is really fascinating, right?
But there has been no paper yet that what is responsible for like the switching thing.
Awesome. Thank you.
All right, Dick Olmsted, you have a question?
Thanks, Banisha. That was very nice. I enjoyed it too. I also didn't know much about this anatomical structure of nectaries. But my question is, what is known about floral nectaries and the sister group Convolvulaceae?
Convolvulaceae, yeah. The
I couldn't see
convolvulaceae,
if they were in the CRC phylogeny or not, and so.
yeah, they are in the core eudicot clades, the rosettes, the stamen-associated nectaries, and the scarpal associated nectaries. They both come under CRC. CRC is regulating this clade whole. Rosettes as well as asteroids in Convolvulaceae family, which is sister to this, because when I was like writing my paper and I had a question from reviewers, so how could you say that two-lobe nectar is the most basal most form? So I had an hypomia by that time, flood. So I took the flower and tried to understand the structure. I found the two-lobe kind of a structure in hypomia. So I feel that this is this lobe thing is like very early in this clade. So if we will see the other Convolvulaceae members, we would find the similar observations that I have seen in Solanaceae.
Thank you.
All right. Not seeing any other questions. If anybody has a last minute question, raise your hand or something. If not, all right. Well, thanks again, Banisha. That was a really cool talk, and I'll go ahead and get the recording uploaded onto the YouTube channel later, so that'll be available. And next.
Thank you so much. And I want to convey that I have been reading Olmsted papers and Stacy's papers for the last 5 years. And you made my work easy because there was an already published phylogeny so that I could trait evolution. And Stacy because I think she reviewed my paper.
We are going to have a time change in the United States between now and the next seminar. So we'll send out a notification just to keep that all straight, because it always makes things a little confusing. But yeah, until then, we'll see you all next time.
Thank you. Thank you. Thank you so much for giving me a chance to present my work.
Oh yeah, thank you for presenting. It was great. We really appreciate it.